Okay, welcome everyone. It's just a few words to introduce our guest. So it's a great pleasure to welcome and introduce Professor Erwin Frey. Erwin Frey is currently chair of theoretical biophysics at the Ludwig-Maximilians-Universität in Munich, where he has built an internationally recognized research group working at the interface of physics, nanoscience, and biology. After completing his studies and PhD at the Technical University in Munich, Professor Frey carried out postdoctoral research at Harvard University, a formative period that sparked his long-standing interest in complex systems and non-equilibrium phenomena. He later held key academic positions at the Freie Universität Berlin and the Hahn-Meitner Institute before joining LMU Munich in 2005 as a full professor. His exceptional contributions have been recognized with the 2024 Max Planck Medal and a recent ERC Advanced Grant in 2023. Professor Frey's scientific contributions are remarkable for both their depth and diversity. His research has illuminated our understanding of complex biological systems, at least in three key areas. First, in active matter and cytoskeletal networks, he discovered how mixtures of particles with different diffusion constants exhibit phase separation, now linked to DNA organization in cell nuclei. Second, he developed a novel theory of pattern formation beyond the Turing mechanism, which explains the self-organization of proteins that guide bacterial cell division, challenging traditional paradigms in reaction diffusion systems. He explained, for example, why some proteins in E. coli spontaneously self-organize in oscillations between the poles of bacteria. Third, he has identified optimal control strategies for self-assembly processes, revealing key aspects of time efficiency with profound implications for both biology and nanotechnology. A hallmark of Professor Frey's career is his ability to move across disciplines, pioneering new directions in which he is able to connect fundamental physics with biological phenomena. He showed how the law of mass action breaks down in coagulation processes and established unexpected connections between evolutionary game theory and quantum condensation. With hundreds of publications in top-tier journals and many thousand citations, Professor Frey's influence on the field continues to grow. He has mentored about 100 PhD students, more or less, right? Many of whom have gone on to successful careers in academia and industry. So today, Professor Frey will share with us his latest insights into the emergence of structure, order, and information in biological systems with his talk, Emergence and Self-Organization in Biological Systems. So please join me in giving him a very warm welcome.
Thanks a lot. This was a very kind introduction. Work? No? Now working?
Yes. No.
Okay.
Yes.
Good. Thanks a lot for this very kind introduction. I'm very happy to be here and give this presentation. I hope I can entertain you in the next 45 minutes and share with you my excitement about this field of research, where I phrased this question: can physics really explain complex systems? Meaning mainly, in my case, can it explain the complexity of biological phenomena that we see? And it's not only me that is fascinated about biology. There's one, I guess you all know, that's very fascinated about biology. Where does it go? Which is Erwin Schrödinger. And Erwin Schrödinger, he wrote this very famous book, What is Life?, at a time where he had to leave Germany for obvious reasons at that time. And he gave a series of public lectures where he tried to make sense out of the complexity of biology. And then he says, as you can read, that of course, if you look into living systems, they will not create new physics because they will be based on the laws of physics. But if you look at those systems, there will be likely other laws of physics, which then, once you know them, once they have been revealed, will be in the very same way an integral part of science as all the other laws that we know it. So let me start with that and show you some of those new laws. In the first slide, I would like to ask the following question, namely, what is the starting point of doing theory in biology? It cannot be elementary particles. It cannot be atoms. It cannot really be small molecules. It must be those kind of building blocks that really make up the cell. And there are a few which are quite important. I have some pictures here. You have proteins. These proteins define the structure of cells. They make filaments. They are the workhorses. They provide motor actions. They drive chemical reactions. Of course, you also have the DNA, and the DNA is responsible for storing information and then reading off the information by producing proteins. And last but not least, there is lipids, and these lipids have the property that they are amphiphilic, and that means they hate and love water, and by that, they organize in bilayers, and those bilayers then form these hollow shells called vesicles, and these vesicles are the containers in which you have cells, in which you also have smaller compartments within cells, and they then organize the chemical reactions and give rise to the complexity of a cellular system. What is the world like at a cellular scale? Let me tell you an anecdote. This anecdote is an anecdote that Charles Darwin wrote down when he was discussing with Robert Brown. And what he says here, before he went on his voyage on the Beagle, which had this famous impact that we all know, he met with Brown, a botanist at the time, and he asked him to look at his microscope and tell him what he's seeing. And then he asked him what it was that I had seen, but he replied, "That's my little secret." And this little secret is what we all know as Brownian motion, and here it is. That's Brownian motion, and that's what's going on. That's what the world looks like at the scale of a cell. So you have this incessant motion that constantly takes place, and then you wonder, why in the presence of such disorder that you see here, can you even create function or organized behavior? Well, there's something else that's quite important. When you look into a cell, there is a energy production machinery that's called ATP hydrolysis, where you have the transformation of adenosine diphosphate, so with two phosphates, to triphosphates, back and forth, and that produces all the energy that we need as living organisms to stay alive. Just every day, every person here in the room has a turnover of 25 kilograms of ATP. And that's a local energy source that's quite key. It's not an external gradient of temperature or gradient of wind or something like that. It's a local internal energy source that every protein can tap into and perform action. So what does it drive? It drives motion of molecular motors. This is a famous movie showing how a kinesin motor attaches to a molecular track called a microtubule by reshuffling ATP binding on its heads, and these heads are feet actually that move along a microtubule. That's driving muscle motion, it's driving cell division, it's really the basic force behind force generation in the cell. It's also something people discuss a lot in the context of active matter systems. That's not all. These ATP energy sources are also used to drive chemical reactions to create order and organization. So here you see a starfish oocyte, and on this oocyte you see this green pattern that runs along the oocyte, and concomitant, as you see, with the wave that runs along the oocyte, you have deformation of the boundaries. And that's beautiful physics because it combines differential geometry with pattern formation, so with non-equilibrium dynamics. Now, there's two things which have been keeping me busy over the last years. One is the self-assembly of biomolecular structures. I will not really talk about this much, but only a little, a note on that. Self-assembly processes, if you look at them, they are a program where you design certain molecules that act in a certain way and then assemble a complex structure like a flagella or ribosome or any kind of other complex structure in biology. And you can compare these kind of assembly processes with a computer program. And as many of you might know, in a computer program, the real task is to figure out how do you design an algorithm to make it fast, so to have a small computation time given the size of the program, and that's called computational complexity. Here, when you look at self-assembly processes in biology, what you're asking is what's the time complexity of self-assembly? So how does the time to finish a structure scale with the system size? And that's quite important because during the lifetime of an organism, it has, or a cell even, they have to create these structures pretty fast because they need them to be alive and to perform all the complicated cellular processes. What I will focus on is the second question here, which is pattern formation in a cell. So I will talk about how proteins organize in the cell and how they guide biological function. Now let me start with a quote. This is from a famous philosopher, Immanuel Kant, who was saying that two things fill his mind with ever new increasing reverence and awe, the more he reflects upon them. Guess what he said? He said, the starry heavens above me and the moral law within me. And I'm sure many of you subscribe to that. I do, to both. Because if you look at the stars, if you look at our universe, it's beautiful, it's wonderful. I would like to change this quote a bit, so allow me that. I would say for me personally, it's the living nature around me and the moral law within me that fascinate me and that fill me with reverence and awe. And why they fill me with reverence and awe, I would like to illustrate with a few movies from real living systems that show you the beauty, the complexity of these phenomena. This is a yeast cell. These yeast cells have a task. They need to divide to create offsprings. And the way they divide is in a two-stage process. First, they need to know, where should I divide? Where should I make a bud? For that, they have a pattern-forming system that creates out of a random arrangement of proteins a polar cap, as you see here on the top. And once you have this polar cap, behind this polar cap, you organize polymers, rigid sticks basically. They bundle up, and there's molecular motors that then push these filaments against the cell wall, and by that, you create a bud site a new offspring, a new yeast cell. There's other processes in cells that are not creating a polar cap, but as you see here in E. coli, you have oscillations of proteins back and forth of a 1 to 2 micron long cell. All the time, you know, all the 20 minutes this cell is living, it's doing this oscillation back and forth. The reason it's doing that, it needs to know where is my midplane. So if you look at this pattern, if you just average over time, you will have a density profile which is maximum at the boundaries and minimum in the middle. So it knows where to divide, and there then it creates a actin ring, FTZ ring, sorry, and this then pinches off the E. coli cell into 2 equally sized daughter cells. I showed you that already. This is the starfish oocyte, where things like pattern formation and morphology changes combine. And last, this here is not a cell moving. It looks like a cell on a substrate, right? But this substrate here is just proteins that are taken out of E. coli and make this foam-like pattern. And this purple object that you see is a hollow shell. A hollow shell just made out of a lipid bilayer. There's nothing in it. There's no motors in it. There's no skeleton in it. It's just driven by the interaction with the foam-like pattern on the surface that you see. And that means that we understand in the meantime these biological processes insofar that we actually can construct these systems bottom up to have lifelike properties and behavior. So now you have seen why I'm in reference in awe about these beautiful biological systems, and now let's dig down a little deeper to understand the underlying molecular mechanisms that drive the beauty of these phenomena. You've seen that. These are the oscillations in a bacterial cell. And what's surprising is that only 2 proteins, they have names called min-D and min-E, are the players here. They drive the oscillation. The way they do that is not particular to an E. coli cell. It's actually a universal scheme by which all organisms drive pattern formation. The design principles are roughly like that. So here you have the membrane, the 2-dimensional surface surrounding the E. coli cell, and the cytosol, that means the interior of the cell. Now you have one protein, that min-D protein. That's this protein which performs the ATP hydrolysis that I was talking about at the very beginning of my talk. So by this hydrolysis, the min protein, the min-D protein, is going from an inactive state into an active state. And only if it is in this active state can it actually bind to the membrane. So it does. As you see here, it binds to the membrane. Once it's bound there on the membrane, there's some nonlinear mechanisms, which are too complicated to explain here, that create a feedback mechanism such that more and more of those proteins are shuffled from the interior to the membrane. Right? If you just would have this path, then what you would do is you would transfer all the molecules, all the proteins from the cytosol onto the membrane, and that's it. No oscillation. You need some kind of antagonist that counteracts the attachment of min proteins to the membrane. So nature here has designed a second player called min-E. It's also recruited to the membrane, as you see here. It's forming a dimer, and in this dimeric state, the min-E is hydrolyzing the min-D, making it inactive, so both of them fall off, as you see here. And then you have a cycle going in circles between different protein states, between a surface and a bulk, back and forth. And these kind of cycles here are a hallmark of non-equilibrium systems. In thermodynamic equilibrium, you have detailed balance. Detailed balance means along all the different connections between the states that you see here, the fluxes have to cancel off. This is not the case here because you drive the reaction by ATP hydrolysis, so you inject a current that drives the system in cycles. And that's important. Without that cycle, there would be no pattern, there would be no non-trivial pattern state. As I said, this cycle here for the min proteins is just the simplest you can write down. This is for budding yeast. Never mind looking at this in detail, but it has the same kind of features, shuffling between states. It has a ATP hydrolysis at its core, and it's going between the membrane and the cytosol. So they all have a unique universal design, and that means there's hope to understand the principles that are behind pattern formation in biology. But of course, I don't want to bore you with all the details of biochemistry here. I would like to follow an advice by this gentleman here to make things as simple as possible, but not simpler. So what would that be? Well, this is the yeast cell as you saw it. You can put this complicated network on a computer, do some finite element simulations for the PDEs, and shwoop, you get the same kind of behavior as the one you see in the data. But as said, we want to make things simple. Now, how simple can it get? And that's not conceptual. I do this not because I want to explain a specific biological phenomenon. I do this because I want to illustrate to you the basic underlying principles behind protein pattern formation. So it's going to be simple and not really quantitative, but qualitative what happens. So let's say we just have one protein, and this one protein can only be in two states. One is it's bound to the membrane, and the other one is in the cytosol. So two-state model. And this two-state model, you can then put into equations and make it even, say, one-dimensional. And what you get then is these two equations saying the temporal change of membrane density is some reactions F plus diffusion. The temporal change of cytosolic density is another set of reactions plus diffusion. And that set of equations is very famous. It was written down in 1952 by Alan Turing in a paper which he called The Chemical Basis of Morphogenesis. Quite an influential paper because it kicked off a whole series of theoretical, especially mathematical papers studying how diffusion reaction systems can give rise to pattern formation. And that's why these things are also called Turing patterns sometimes. But in a way, this equation here is not quite the right starting point for the system we are interested in. As you saw from this reaction scheme, an essential feature of the reaction scheme was that no protein was lost. It was recycled. It was going from one state to the other, but never decayed or was never produced. So that equation should respect mass conservation. It doesn't because it's based on two functions that you see here. You need to modify it. And the way you modify it, you say this function g must be minus f because otherwise the reactions don't really balance and you wouldn't have mass conservation. Right? So as you now add up the two equations, let me do that, you get an equation like so, where the temporal change of the total density n plus c, is given by some diffusion terms, and you can write this as a constant times the Laplacian of a potential that we call eta. So for those of you that know something about critical dynamics, critical phenomena, this is a famous structure that's known in the critical dynamics of liquids. It's known as the Kahn-Hiller model, where the eta that you have here is the functional derivative of a free energy functional and driving then the motion of these critical phenomena. But that's not an equilibrium system here, so this eta is not a potential. It's not a thermodynamic potential. It actually is a dynamic quantity in its own right. So it obeys an equation of motion which has this structure here. So there is a link to known physics in critical phenomena, but it's different because this system is off equilibrium and there's no free energy functional that you can rely on. Now that's the math, and now let me give you a geometric analysis that alludes to the key physics that's going on here. So for that, let's assume we are in a well-mixed compartment where diffusion is super fast and the only thing that happens here is reactions. And let's say our reaction function F is decomposed in attachment processes A of M and detachment processes D of M. The steady state then of this well-mixed dynamics is very simple. It's two ordinary differential equations with two parameters, and you can analyze these ordinary differential equations in a very simple way, namely by looking in phase space. Well, students may know phase space from classical physics, where you have phase space analysis of harmonic oscillators, which have Hamiltonian functions that you can write down and analyze the path of the trajectory. Here it's a bit different because those are dissipative systems. Energy is not conserved here, it's dissipated in the environment. But still, you can use the phase plane to analyze the system, and there's one characteristic line that you have to draw, and this line that you have to draw is F equal to 0, because F equal to 0 means that all the partial derivatives with respect to time are 0, meaning the system is in a steady state or in an equilibrium state. Not a thermal equilibrium state, but a reactive equilibrium state. So this here is the manifold or the line characterizing all the possible equilibrium points. And the system is basically free to choose one of those equilibrium points, but not quite, right? If you look at our system, it has mass conservation, and because it has mass conservation, it needs to lie on a simplex like so, where m plus c is n, and on this line, it can only then react, and then by reactions, be driven towards the fixed point, which is the intersection of this mass-conserving plane and the line of equilibrium. So now, as you have that single point, one point that you need to realize here in mass-conserving systems is that as you change the mass, you're changing the plane in which those reactions takes place. So this total density of particles that you have becomes a control parameter. It seems trivial, but it's very, very important. If you think about the physics of phase transitions, you have a temperature that you tune, and as you tune the temperature low enough to the critical point, you will have a phase transition, and magnetization will emerge, or you get a superconductivity and other kinds of order parameters. But temperature is fixed. You fix it externally, and then it's just constant. Here you have a control parameter. Because it's a density, it can vary in space, and it can vary in time, and it's dynamic. So imagine a system showing phase transitions where temperature is a local dynamic variable. That's how you should think about it. So now once you have understood that, so that you have local equilibria, and these local masses between different compartments are exchanged by diffusion, we can argue why there are oscillations or why there is polar patterns in a E. coli cell, simply by saying, for the sake of argument, let's take a two-compartment model, a left and right domain for that. And let's say there is a diffusive exchange between the two compartments happening mainly in the cytosol, not on the membrane, just for simplicity. So as you start the argument, you start from a single point, here this gray spot, where the densities on the left and the right are equal. Now let's add a perturbation. So let's say I put a little more mass to the right and a little less correspondingly to the left. So I will have these two lines now defining the reaction planes for the left and the right compartment. But now reactions kick in, right? The reactions will pull you towards the set of equilibrium points. So they will pull you up on the left, and they will pull you down on the right. And as you do so, if you now look at the axis of the cytosol, you will have a gradient. Higher cytosolic concentration on the left compared to the right. A gradient in density obviously needs to give rise to a flux by Fick's law. So we will have a flux by Fick's law. This flux by Fick's law increases the density on the right even further, and it lowers it correspondingly on the left. So what you have seen now is a self-organized amplification mechanism by which small perturbations grow. And now if you look at the argument, what I've basically argued is because the slope here is negative, you have an instability. If you turn the slope the other way around, you don't get an instability because all my arguments reverse and the system goes back to a homogeneous steady state. So what you have obtained from this simple geometric argument is a geometric criterion for the existence of an instability. You can all do all this analytically, of course, but the beauty here is that the geometry of an object in phase space tells you everything you have to need about the instability of the system. And that really is the heart of these systems showing pattern formation. That's the basic mechanism giving rise to polar patterns in cells. Now, let me show you how this works out in a computer simulation. So here you have your phase space with the null-Klein, the mass-conserving plane, and here's some initial distribution of points. As you can also see in real space here, the membrane density in physical space and the cytosolic density also in physical space. And as I start now the simulation, the initial perturbations grow. They grow into patterns, as you see. Then in phase plane, it converges to an attractor, this line. And on this attractor, the dynamics doesn't stop. There's a slow dynamics continuing, as you see. And in the long run, there was coarsening, and you have the development of a polar state with a low density on the left and a high density on the right. Okay? Now, what have we learned from that? I think the main message I want to convey to you is that you can rephrase differential equation in terms of geometry. That's not a new thought. Poincaré, when he was thinking about how to analyze nonlinear equations in mathematics, his statement was we need to know the geometry of the flows and not the analytic solutions, because you can't get analytic solutions of these nonlinear equations. So it's a geometric picture here of partial differential equations, and it helps us to answer two very, very basic questions about pattern-forming systems in biology. One is, why do proteins self-organize into concentration patterns? And the second one is, what determines the properties of these patterns? The answer to the first question is a geometric answer, the slope criterion that I explained to you in the simplest possible terms I could. Actually, if you go beyond that, you can develop a full geometric classification scheme by looking for, when you have a multi-component system, at the manifold, the high-dimensional manifold of the equilibrium points, and then deriving geometric criteria on the slopes of these manifolds, and thereby obtain, again, criteria for instability of non-equilibrium steady states. Second question, the answer is these local equilibria I showed you the system is pulled towards as it was competing with the diffusion in the system. And you can again develop this into a full-blown theory of local equilibria where mass transport is kind of the guiding field that gives rise to different bifurcations in your system, and that's then followed by the dynamics of the individual components. So this is as simple as I can put it. If you like to hear more, students, also faculty, if you like, are welcome to listen to my lectures. They are on YouTube now, and I just started last week a course on self-organization and pattern formation where we talk about these things much more in depth than I can possibly do it here. So let me continue here with more applied things, because when you say biology, you better mean biology when you do biological physics. You can't just do this as an excuse to do some models. You should be able to actually explain real biological phenomena and processes. So can we explain actual biology? And as you saw, again, there was this equivalence of phenomena where you showed oscillations in E. coli cells, and we can reconstitute them in a computer and see the same kind of oscillations. That's not a big deal, because you can actually design a lot of mathematical models that show similar phenomena. If you want to be able to compare with biology, and actually, in general, if you want to make a theory, the real test of theory is comparing with experimental data in a quantitative fashion such that you, as a theoretician, are able to predict, and the experiment is able to falsify what you say. Well, here, that's the molecular scale. There are these proteins, the min-D proteins and the min-E proteins. I actually was now telling you that the min-E protein also is a switch, and you have one switch switching the other one back and forth. And now we have to see whether we can have an actual theoretical prediction. And that's a very long story. So for those who don't know the field, not only in high-energy physics, experiments take long. In biology, experiments take also a very, very long time, because it's complicated to design genetic tools in order to be able to engineer the protein content in the system. You really have to work with a living organism to make a phase diagram, and that's hard and that's difficult. And I was chatting with Suk-Yun Yoon from UCSD 7 years ago to convince him to make an experiment in a living organism, an E. coli cell, to test our theory. And from that, and that was just published very recently, we were able to create an in vivo phase diagram. They were able to, not me. And what they found is that they could really plot out the phase diagram as a function of the concentration of the protein min-E and min-D in micrometer per cubed, so in real units. And here, this colored region that you see is where you have pattern formation. Before I talk about the theory, let me mention a few things here about the biology. The wild type, so the actual E. coli cell in its natural environment, is right here on the bottom. That's kind of interesting, because it means evolution has engineered the rates such that the amount of min-E and min-D you need to produce is minimal. Otherwise, you would waste resources to make more. So it's not over there. It's really here on the bottom of stability. And this point here, however, is still far enough away from the boundary of stability such that the system is stable to fluctuations, which you need, because E. coli cells, after all, live, when they're in your body under 40 degrees, and when they're outside your body at 20 degrees, so there's quite some harsh change in environment. So you have to have resource efficiency, and you have to have a robustness against perturbation, and that's valid here as well. Now here there's a comparison with theory. It's a comparison in units, right? This is the same micrometer cubed, that you have here for the concentrations, and you find basically the same kind of phase diagram, the same region, not only the same phase diagram, the same type of modes, standing wave patterns versus traveling wave patterns here in the experiment and here in the theory as well. And if you dig even deeper, you would find that the wavelength, which is important for biological function of E. coli cells, is exactly the same in the theory and in the experiment. So we really, after many, many years of research and interacting with biologists, found a quantitative model that's falsifiable. So are there new laws? This question of Schrödinger, and I would say, yes, there are quite a few new laws that you find. One is this geometric classification scheme I showed you. Another one is that these protein patterns are able to sense geometry. So here's a wedge geometry, and here's the pattern of these min proteins in such a wedge geometry. It's a crazy pattern, right? It starts with spiral waves and some chaos here behind, and this chaos then, like a flame front, invades the regime where you have spiral waves. And after a while, it seems like this all is vanishing in chaos and very disorganized structures. But after a while, as you see here, another phase of pattern formation kicks in, and you have the formation of these line-like structures, very much like smectic liquid crystals with all kinds of topological defects that are floating around here in this final steady state. And this is all driven by just putting a wedge on the pattern-forming system. And finally, I think that's also a pretty deep insight into the physics and biology of these systems. We know now that, oh, what happened? We know now that function in these systems is modular. There are emergent features which are quite important. One is the ability of these systems to self-organize. The other one is to be able to be robust. And the third one is to have emergent length and time scales, like the clocks that you saw of the filament ticking back and forth, and the length scale that's necessary to actually create proper and faithful cell division. And that's encoded here in different modules that you see here in the reaction network. So the outer network is the pattern-forming module. This here is the robustness module. And if you include that here, you get the module responsible for the emergence of length and time in these systems. There's other laws you might know. There's thermodynamic laws. And you can phrase them in different ways. I want to look here at line tension. I guess you all know what line tension is. Line tension is the tendency of interfaces to become shorter. In a liquid gas system, the line tension is a consequence of pairwise molecular interactions between particles in a very simple way. If you have something here at an outward bending interface, it is energetically unfavorable because it's just missing interaction partners. If you, on the other hand, bend it inward, you have more attractive interactions. So there's an energy mismatch or free energy mismatch between those two configurations, and that's why this interface straightens out. Right? So the molecular basis of line tension in this case is pairwise interactions. When you formulate then the thermodynamics of that, that's a famous law called the Gibbs-Thompson law. It doesn't know about these molecular details. All it knows about is there's a density jump delta rho between high and low density. There is some curvature kappa along the interface. And this then gives rise to a surface tension which is driven by gradients in a chemical potential, because those chemical potentials then drive fluxes, and these fluxes then straighten out the interface. Okay? So this law here is agnostic about the molecular origin of surface tension. And it has implications. So for example, I like to show that slide because it's actually an engineer at the TU Munich some time ago, and he asked his students to make different kinds of structures and then put soap on it and then beautifully see these minimal surfaces. Right? There's a beautiful structures, and some of you might know that they were actually used, those ideas, to construct the tent of the Olympic Stadium in Munich. That's not what I wanted to say, but I just find it very cute. So is there surface tension in cells? Is there surface tension in non-equilibrium pattern-forming systems? And my answer here is yes, and I'll prove it to you. So here you have another system. It's a C. elegans zygote, and it needs to form a polar pattern. So you can simulate a reaction network, and when you now look at the movie of how these patterns form in the C. elegans zygote in a prolate and oblate geometry, if you looked at that movie, it changed to a configuration where the length of the interface is minimal, right? That's the minimal length, not this one. And this is the minimal length, not the one on the equator. So there is a tendency of interfaces between patterns to minimize. Now the point is, diffusion reaction systems are built on a model that has no basic physics interaction in it. It has reactions, but no pairwise interactions via van der Waals forces or something like that. So the mechanism that gives rise to Gibbs-Thompson relation in the thermodynamic example I showed you is not the same. But still, there is a surface tension. So this object surface tension is a property whose microscopic origin can be manifold. It doesn't have to be pairwise interactions. And what we found out in this paper with Henrik Weyer and Tobias Roth that is on the archive is, and I don't have time to explain that there is a surface tension in pattern-forming systems, but it's based on non-equilibrium fluxes and not forces. It's based on the fluxes I showed you at the very beginning of my talk, where proteins go from the membrane to the cytosol and back. Those fluxes drive the system in a state with an interface that has the property of a surface tension. And if it has the properties of a surface tension, then you should be able, in these pattern-forming systems, to observe structures which otherwise in nature you find for liquid foams. And I guess you know what kind of, for systems with line tension, and one of them is a foam, the other one is multi-component mixtures, and in particular, right, I said it already, it should show foams. And then, when we started to understand that, we digged into early literature, also from the group of Petraschwili in Munich from 2019, where they showed in one of their figures this beautiful picture of a labyrinth-like structure. And that looks like a foam, right? But if it's a foam, it should obey the foam laws. And there's quite a set of foam laws that one knows in studying liquid foams. One is Plato's law, meaning there should be 120-degree angles at the vertices connecting the boundaries. So here, at such a vertex that you have here, the angle should be 120 degrees. You can check that. So Henrik and Tobias did. And indeed, there's a very narrow distribution of 120-degree angles in such a foam. So from that perspective, it's geometrically a foam. It obeys the Plato laws. If you look further at the size of the cells, it has a finite size distribution, and that's distinct from a liquid foam. As we all know when we drink beer, right? The beer foam vanishes after a while, so it dries out. That's not the case here. These kind of foams, they stay and have an equilibrium distribution of foam sizes. So that's distinct and needs an explanation. And we can study that because there was just this picture here in the paper. There was nothing else. So we could then use the computer simulations, since we have a valid, biologically tested, predictive, quantitative model. So we can check what's going on in these systems by doing computer simulations. And you can do beautiful experiments with computers because you can recapitulate an experiment under very controlled condition, which is very hard to achieve in an actual experiment. So what Hendrik did here is he took the equilibrated foam-like structure that you now get from the computational model and sized it down, made it smaller. So if you now start the dynamics, it should go back to the equilibrium steady state by growing the cell sizes. But you have to know how do those cell sizes grow. You know, what happens here? There's cells here, as you can look at them. Some have 5 edges, some have 6 edges, others might have 7 edges, so they have different kinds of topology that you have here. The movie goes a bit fast, so watch carefully. So it coarsens, so it goes back to the equilibrium state. And it does so in a very interesting way. It does so by the cells with the 5 edges, 7 edges to grow, the ones with the 6 to stay constant, and the ones with the 5 to shrink. And that's also what you see in foams. And the reason you see this in foams, and that's, I think, very spectacular, is that this is based on surface tension alone, and the formula for Neumann's law follows from the existence of a surface tension and a very famous theorem from differential geometry, which is the Gauss-Bonnet theorem. And the Gauss-Bonnet theorem, just, you use it for a system with surface tension, and out you get the von Neumann law. And you see it's valid here. So another quite universal phenomena that requires no more than the existence of a surface tension and geometry. You can do the opposite experiment on a computer. You can scale up the system and make it larger than it originally was. So let's scale it up. And as you scale it up, it has to shrink down. And it shrinks down by topological transitions. So the topology of this network is changed by, as you saw, events that change the connectedness by doing something like a cell division in these systems. So both of it, the coarsening implied by surface tension and von Neumann law, and the reverse transitions driven by some surface instabilities, drive the steady state with a homogeneous, with a stationary distribution of sizes. Now, once he saw that, we asked Petra and her students, did you do more than just this one snapshot? Don't you have something in your files which you could show us that shows some movies of things that you have been observing at that point in time? And they found some. And let me show that. These movies show that the 5 shrink and that you have these kind of division events. And I'm looking forward to many, many more experiments where people can check in detail the predictions Hendrik made in his theory about what we call now Turing foams. Now, let me conclude with a crazy question. Can we build lifelike systems from the bottom up? And can we, as theoreticians, help? Or is this an experimental question? Well, I wouldn't ask the question if I wouldn't think we have an answer. And it also follows a line of thought Richard Feynman had in a different context. He was kind of saying that if you can't really construct something from the bottom up, you don't really get it. You don't really understand it. And building a cell from the bottom up is really very helpful in understanding the basic mechanisms that underlie living systems. After all, for us humans, it's very, very hard to think in terms of 100-component systems with all kinds of regulatory mechanisms. We need to build our intuition from simple to more complex. Why do I think so? I think so because the most promising system for me is very simple. It just consists of a hollow shell of a lipid bilayer, a vesicle, in which you have proteins, and those proteins can float freely or attach to the membrane and thereby change the mean or Gaussian curvature of these manifolds. And then you have everything together that gives rise to complex phenomena. You have proteins through mechanochemical coupling can deform the geometry and change the membrane and its geometry. And as I showed you, these protein patterns are geometry sensing. So as you change the geometry, the pattern will change. And then you have a feedback between the two. A change in pattern will change the morphology. A change in morphology will change the pattern. That this works, we have shown with Petra. So here, I again repeat this movie. Here's a cartoon. To orient yourself, these are the foam-like patterns that I explained to you on the basis of non-equilibrium surface tension. And on that foam-like pattern, you have this vesicle here, a hollow lipid vesicle. And now, as we start the experiment, this moves in a cell-like fashion. We can reconstitute that computationally. That's work by Jan Willeke in the group, who just started a year ago. And here he simulates with phase field methods, solving the equations that also these objects move in a cell-like fashion. So let me conclude my talk. I hope I could show you that by seriously thinking about biological problems, new theory opens up. This new theory is really rethinking how we do theory of pattern formation, and this has helped us to understand that local equilibria, together with a mass redistribution instability, are the basic physical mechanisms driving pattern formation in cells. This theory can be applied to real-life problems. It explains in vivo patterns. It gives rise to new non-equilibrium laws like surface tension that give rise to foams in pattern-forming systems. And it can explain cell-like motion in synthetic cell experiments. Where do we go from here? Well, the future looks bright, I would say. Bright in many, many ways. Combining the physics of pattern formation with the physics of dynamic manifolds in terms of differential geometry and non-equilibrium physics is a beautiful new route to go. And this last system to help engineer biotechnology based on insights from theoretical physics is also, I think, a super promising direction. Here's the people who did the work. These are the students that worked on it over the years. Friedhof and Jakob, they started this whole thing with me, designing the local equilibria theory. Jan, you saw this was the simulation. Henrik and Tobias did the work on the Turing foams. Leshy was the one working on the geometry sensing, and Tom and Manon worked on the starfish oocytes. And these are my heroes in the experimental arena, Petra, Nikta, Kees, Sukyun, and Lidevai. And I'm really always very, very happy to interact with experimental colleagues because they stimulate our thought, and very importantly, they keep us honest as theoreticians because we have to compare with real-life data. Thanks for your attention.
Thank you. Thank you very much for this very nice, stimulating talk. So we have time for questions, even naive questions.
Anyone.
Any question?
So you have shown that microscopic laws can, or microscopic processes, not laws, can lead to pattern formation. But what is the advantages from biological point of view to form patterns? Do you understand that?
Yeah. So patterns are super crucial because from this first experiments I showed you, without the polar cap, the actin filaments wouldn't know where to go. Without the min patterns, the FTZ ring wouldn't know where to go. So patterns are the rulers in biology. They determine length and time scales, and they organize cellular activity. So they're really crucial. They are functional and central to our understanding of living systems. And it motivated actually Turing to start out with this 1952 paper because he was thinking about chemistry guiding morphogenesis. And morphogenesis is an even more complex problem where you have tissue and this tissue is reorganizing and you have folding and really the development of an embryo, which is also guided by genetic patterns and protein patterns and morphogens and all of that. So I think patterns in biology are not stripes of zebras, right, or leopards that have some points on their furrow. It's about intracellular organization where biology really has a lot of important function. Yeah.
Come on, don't be shy.
Yeah, I don't bite.
Thanks a lot for the very nice presentation. So I had a couple of questions. Maybe just stick to one. So you are essentially pointing your attention and just looking at these proteins, even though there's a lot of stuff around that is interacting and is reacting. So you are just essentially saying that the mass conservation concerns just these two species. What about the rest? For example, just ATP, which is the driving. Usually it's assumed to be buffered in cells. Is it really true? Can it be that it's depleted, and so we should have to consider other species into your equations, such that so we have to use the mass conservation also for other species? And how you mention something about multidimensional systems? How does this theory work in this case?
So, of course, you're asking a very important question. Let me answer them step by step. ATP in a cell is abundant. 25 kilos a day, right? There's a lot of turnover, and it seems that biology is very wasteful with ATP hydrolysis, so this is not depleting. There's always enough there. You can construct experimental situations where you deplete the ATP, and of course then this thing starts to stop, but not in the cell. Now, concerning other components, for the Min system, of course, we worried. I mean, is this the only thing? Are there just two proteins, and do they do the job? What you can do there is you can actually, and that's quite amazing, do reconstitution experiments, meaning you take the two proteins, you purify them, you put them out of the cell, you put them in a liquid environment, and then you know there's just those two proteins. And you can apply your theory to the system where you know that there are just two proteins. And we were lucky, same theory applies. So it's a complete theory for this Min system. Now, when I go to yeast, where we showed this very complicated network, here, maybe I go back so everybody can see it. So here, this is complicated, right? This is on the right, there's lots of different proteins. And it's not even clear that you know all of them, right? And this is a very general problem in the theory of complex systems, especially in living systems, that you don't know all the parameters that determine your system. So it's quite an endeavor each time you study those systems to figure this out in close interaction with biology and biochemistry, physics and biophysics, etc. So let me answer conceptually. If there are proteins that are produced and degraded That's fine. That's not a problem, because here in the yeast cell they are, but the approximation of having a constant protein density is the better reference frame. It's the better starting point, because approximately this is always true. What the cell then does is in the yeast cell, as in contrast to the E. coli cell, it not always has a polar cap. Most of the time it doesn't have anything, because it doesn't want to recruit all the actin filaments every time. So there's a cell cycle, and in this cell cycle, there's a certain point in time where the cell starts producing, it's not on there, a certain protein, and this protein then drives the reaction network through a bifurcation point, and then the polar cap formation starts. So you can even utilize the fact that the total density is a control parameter to gear, control these kind of pattern-forming systems. Hope that answers your question.
Yes, thanks.
Thank you very much for the talk. So at you were showing this function in terms of the different densities, I think, of the membrane and within the cell and the bulk, and with the negative derivative. And you were saying that the reason why you could see that it's a periodicity in this non-equilibrium physics is because of this negative derivative somehow, and that you said that this is just an example that you could generalize it in terms of differential geometry, like language. Can you say more about this? And yeah, I would like to understand more about it.
Okay, this is a longer story. So what you do is you look at these diffusion reaction systems. So you write down this whole set of differential equations, and then you formulate a condition for linear instability in these systems. And that's related to the Jacobian of the reaction terms, okay? And once you have the Jacobian is the Jacobian on this manifold of equilibria, right? And then what you get out of this is conditions on the Jacobian on this manifold, and that then gives you information on the stability and instability.
When you say the manifold, it's the manifold that characterizes all of your system, right? So at the given point in T.
Think about the manifold as the equilibrium of concentration of min E component 1, 2, 3, 4, and min D concentration 1, 2, 3, 4. And then as a function of the parameters, that gives you a high-dimensional manifold in space.
I would say this is just a point in phase space. It's not a manifold. I cannot see why this is a manifold.
Okay, okay, good point. It's a manifold because the total density of all your variables, min E and min D, is a control parameter, and they span the manifold. So for each density, you have a point, but as you change the densities, you span the manifold. So if you have two densities, you span a manifold on a two-dimensional support, and if you have three densities on the three-dimensional support, etc.
Right. So your phase is a manifold, and you want to look at the evolution in that phase space. Is that correct?
On the equilibrium manifold, not overall in phase space, but on the equilibrium manifold.
Yes. And then
that shape, you don't have to. That's the point. You do not have to know the dynamics of your system to learn about the instability, and that's the advantage, right? If you were to know about the dynamics fully, of course you know when things go unstable, but we try to infer a dynamic phenomena just on the basis of similar like in thermodynamics. Like in thermodynamics, you infer stability and instability from the free energy landscape alone. And here we wanted to have something similar where you infer stability and instability from the set of reactive equilibria alone, which is the equivalent of thermodynamic equilibria. That's fine, yeah.
Thank you. So linking back to the first question, I wanted to ask you, so basically you reckon that evolution led to the rise of such complexity in biological systems so as to accelerate or to allow, let's say, the formation of new life. And can we get to the point of saying that such complexity is a necessary condition to have life in the first place?
You ask a very difficult question. I cannot answer this. It's impossible to answer because, you know, strictly speaking, evolution is a historic theory, right? So we don't really know for sure. It's very hard to tell. But we still do research on this, and we try to uncover the laws by which evolution worked. What I can tell you is something maybe in reverse, which might help you getting an idea of why these simple systems are those that are at the beginning of an evolutionary pathway. So when you look at this system here of the yeast network, it's very complicated, right? It's a higher order organism. It's later in evolution than E. coli. The proteins that you see there, they have different names, and they're not related to the proteins that you have for the E. coli system. So Ludwig-Maximilians, you saw her picture on my last slide, she dared to kill one of the central proteins that you have here in this network, which is this BEM1, just deleted it by mutation, and then looked at the yeast cells over many thousand generations, and they're very robust. And what happened is they regained function within a few thousand generations. Within that time span, you don't have de novo creation of proteins, just not possible. That's too short. But what you can do is you can delete stuff. You can get rid of things. And she could monitor that because in these experiments, you can watch every single part of your yeast cell. You can sequence them so you know what's in there and what's not in there. And she could specify what she lost. And then miraculously, the network that remains is topologically the same as this one. And that I found super amazing, that two systems that are evolutionarily distinct, not the same molecules, boil down to the same mechanism, which is based on physics and not based on evolutionary traces and phylogeny. So I think those things are pretty robust cores of pattern-forming systems, and maybe E. coli and bacteria have the earliest of those pattern-forming systems. That's pure speculation. Yeah.
Thank you. Thank you.
Good afternoon. I have a question about the oscillation of molecules in the bacteria E. coli. How the cell detect the center of the cytosol just from that oscillation? How does the cell know in a molecular way where to send the molecules to complete the division?
So how it works is roughly like so. So you have the oscillation, and then you have high density at the poles. And there's this second set of protein called FTS set. It's a protein that can make rings. And this protein is degraded by the min proteins. So what biology does, it hinders formation of stuff at the poles, and then can only form at midplane. That's how it works.
Okay, thank you.
Yeah. So it's an inhibition mechanism. Yeah.
Good afternoon. When you showed the movie about the cell, the synthetic cell moving on the lattice, could you please take it back from the slide?
Sure. I love the movie. It's wonderful.
What you can see is that the moving of the synthetic cell alters the undergoing lattice in some way. I would like to ask if it's dependent, the movement of the synthetic cell from the lattice is
yeah.
Or if it's the pattern that drives the cell, or the velocity of the cell depends on it, or something like that.
So it's both. it's not really a lattice. So what is the bottom is a lipid bilayer, just a liquid, right? And on that liquid, you have a protein pattern, and those are the green, foam-like structures that you see. And there's also a pattern on the vesicle on the outside. Okay, and now as the cell moves, there's the surface here, and there's a wedge. This is the cell, and here's that wedge. And there's a wedge geometry like the one I showed you when I said there's geometry sensing in patterns. So as you have this wedge geometry and the cell deforms, it will change the pattern underneath, which is the green pattern that you see there. Right? So you have a feedback of the geometry of the cell edge on the pattern, and then the pattern itself are proteins that interact with the other proteins on the vesicle, and that gives a gradient in adhesion that pulls the vesicle forward. Right? So you have this vesicle kind of deform and lean to one side, changing geometry, changing pattern, changing the geometry.
Okay, so that could be used to control the movement of very little organisms or cells like that, with alternating bilayers of a particular pattern or from another?
In principle, you could now engineer the diffusion reaction network to have different length scales, different types of patterns. You could engineer all kinds of things to control the motion of the bacteria along the surface. Yeah.
Thank you very much.
Okay, good. I think that we are delighted to have you here. So let's thank again Professor Frey for this wonderful talk. Very nice indeed. Very clear. Thank you. Very clear. Thank you very much.